Hello, welcome back. Today we're going to answer the question, what was the Human Genome Project? And I'll give you a spoiler alert, it was one of the most important scientific projects of the last 100 years. It's still paying dividends to all of us because now that we understand the structure of the human genome, then we can all benefit from the medical advances that have followed on because of that. Now we're also going to dive into DNA in a fair amount of detail, talk about what the base pairs of the DNA molecule are, and sort of how it fits together so that we can have an appreciation for how incredible this project really was. So go on this journey with me as we learn about the Human Genome Project and the structure of the DNA molecule. The Human Genome Project was an international scientific research project with the goal of determining the base pairs that make up human DNA and of identifying and mapping all of the genes of the human genome from both a physical and a functional standpoint. The project was coordinated by the National Institutes of Health and the U.S. Department of Energy. The project was launched in 1990 and was completed in 2003, having mapped the entire human genome. The project was a massive international collaboration involving researchers from around the world, including the United States, the United Kingdom, France, Germany, Japan, and China. Genes are made up of DNA, which is a long molecule that's made up of a sequence of 4 different nucleotide bases: adenine, cytosine, guanine, and thymine. These 4 nucleotide bases are arranged in a specific order, and it's the sequence of these bases that determine the genetic information contained in a genome. Now let's just take a second to appreciate what this project was actually trying to do. The goal of it is to map the sequence of these nucleotide bases, which I'm going to show you in just a second, in the DNA molecule. Now, to appreciate it, consider that the DNA molecule we now know has 3 billion, that's billion with a B, nucleotide bases. And the goal of the project was to map each and every one of these chemical bases that are in the human genome. By understanding the sequence of the bases, we can then understand the gene structure and sort of understand how the human body is organized from a genetic standpoint, right? We're going to talk about the implications of that as far as genetic medicines and things as we go down through this. What I want to do now is dive in a little bit deeper into the structure of DNA so you understand what a DNA base is. But as we go through this discussion, I want you to consider there's 3 billion bases in every DNA molecule, and every cell in your body has a copy of DNA, multiple copies of DNA in your cells. So it's a very complex molecule in every cell of your body. All right, so we're going to go through this in 4 different sort of levels. We're going to start very, very big picture, and then we're going to get more and more detail, but just keep in mind that everything I'm going to cover here is still considered sort of the basics. I mean, the actual details of how DNA functions is extremely complex and actually still researched today. We don't understand everything about DNA, but everything I'm about to tell you now, we absolutely understand that this is the way it works. So here is the cartoon that you see of the DNA molecule. It's a double helix, which means there are 2 different helixes there interwoven with each other. So there's double helix, right? And this double helix structure, I haven't shown them in this drawing here, but it's called a phosphate backbone. We're going to look at it in a little more detail in a second, but there's a lot of phosphorus in here, and that forms the backbone, the scaffolding, if you want to think of it as a ladder that the DNA is made of. Now there are 4 what we call base pairs. In this drawing, they're just labeled as colors, but in a second we're going to get into the molecular structure of what they are. If you're not an expert in chemistry, don't worry about it. I'm not expecting you to know anything about chemistry, but I want to share with you the beauty of the things that we've learned. It kind of blows my mind what we've been able to understand about ourselves. So even if you don't know chemistry, just take a deep breath and trust me when I say you will be able to understand this, okay? Now, again, they're molecules, but here we're drawing them as little colored pictures. So we have 4 different, what we call nucleotide bases. They're just fancy words for 4 different sort of sub-molecules that make up DNA. One's called adenine. Here in this, it's a kind of a yellowish color. One's called thymine, one's called guanine, and one's called cytosine. All right, now one thing I want to draw your attention to here is that all of the bases, the nucleotide bases, they appear inside the double helix. So they're kind of like the rungs of the ladder connecting one side of the phosphate backbone to the other. But notice that in the middle, you always have 2 different nucleotide pairs. Notice purple here is thymine, and that one's connected to adenine. And then here is sort of a deeper purple here, guanine, connected to the red one, or the pink one called cytosine. And if you look down through here, the purple is always connected to the yellow one here, yellow, purple, purple, yellow, yellow, purple, purple, yellow, yellow, purple, like this. So what we have learned is that adenine, we're going to abbreviate it with the letter A, so we don't have to say adenine the whole time. Adenine is always connected and always bonds to, I'm going to put a little double arrow here, the thymine. Right? So when you read about DNA, you read the A's are connected to the T's. And I'm going to show you exactly why in just a second. Okay, that's the beauty, that's the kind of like the amazing thing that we've learned about DNA is how it's organized at a molecular level. Right? So the A's are always connected to the T's. And then if you notice, everywhere you have a deep purple, it's always connected to pink. Deep purple, pink, deep purple, pink, deep purple, pink, deep purple, pink, and so on. It could go the other way, pink, deep purple, and so on. So we also know that the guanines are connected to the cytosines. So if you had a microscope and you could read, you could actually see the letters of DNA, everywhere that you would see an A on one side of this rung, you know that it has to connect to a T. And everywhere you see a T on this ladder, this double helix, you know it has to be connected across the rungs to an A. They only bond like this. You can never have an adenine connecting to a cytosine, for instance. Right? And similarly down here, the guanine is always bonded to the cytosine, cytosine here. And the cytosine, if you ever see one of those, it's always connected to a guanine. And that's the purpose of these drawings. I'm going to show you why that happens in just a second. So this is the cartoon version that you often see with A's and T's and C's and G's. Now let's go one tiny level deeper. This is not too much deeper, but what we have here is we have the double helix structure, we have the phosphate backbone, the two helices, they're twisted together, and then we have the same thing. Here it's labeled T connected to A, and that's what we had here. A and T are connected together, and then C connected to G, which is what we have written down right here. So this is the same information drawn here in colors. The only difference is now we have spelled out what the actual thymine molecule is, or this little nucleotide base is what it's called. So you can see there's a big structure right here. We'll talk more about it in a second. And then we have the adenine right here. Now the adenine's connected to the thymine, just as we have said up here. But we're zooming in and showing the molecules. Now you don't have to be an organic chemist to appreciate what this is telling you. Let me show you how to read this. When you see these little hexagons like this, every time you see a point, like a corner here, that's a carbon molecule. So there's so much carbon in the human body, we don't actually write all the carbons down. There's carbon everywhere. Instead, what we do is we draw it with these little kinked structures like this, and every time there's a bend, you know there's a carbon here, and the carbon here, there's a carbon here, carbon here, carbon here. Sometimes we write the carbons when they're off of the main ring, like this, in organic chemistry, but in general, when things are kinked with these lines, everywhere there's a bend or a point, there is an understood carbon at that location. So carbon, 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 everywhere you see a bend like this. All right, now you have the similar kind of thing down here. We don't need to dive into the details right here, but what I want you to notice is how they come together, right? This is one half of the DNA molecule, and then this is the other half of the DNA molecule, and they're bonded together in the middle with these little dashed lines right here. A always goes with T, and C always goes with G. Again, we're going to dive into why they only bond this way in just a second. But before we dive one level deeper in the next drawing, I want to call your attention to the fact that the nitrogen, here, there's a nitrogen atom hanging off of this carbon. It is connected to an H, hydrogen. There's a hydrogen here connected to this oxygen. There's a hydrogen here connected to this oxygen. There's an a hydrogen here connected to this nitrogen, and there's an oxygen here connected to this hydrogen. What have you noticed? All of the connection points between the nucleotide bases in the DNA molecule involve hydrogen. All of them. Look right here. Hydrogen connected to oxygen or bonded to oxygen, hydrogen bonded to nitrogen, hydrogen bonded to oxygen, hydrogen bonded to nitrogen, hydrogen bonded to oxygen. This is called hydrogen bonding. You can guess why, because it's a bond, a chemical bond involving hydrogen. Now, hydrogen bonds are important in chemistry because they're strong enough to bond it together. In chemistry, a chemical bond is a force that holds two atoms together, essentially. You can think of H2O being water. There's bonds holding those oxygen and hydrogen atoms together. Same thing here, there's just more atoms that are bonded. But hydrogen bonding is just strong enough to keep it bonded, but also not super strong. Why do you think a DNA molecule right down the middle, because every one of these connection points functions as a zipper, Why do you think it's important that these bonds here, these hydrogen bonds, are not that strong? Well, because DNA needs to be able to separate. That's how the cells multiply and how the DNA molecule copies itself. We're not going to get into the copying mechanism. The copying mechanism for DNA is quite complicated, but basically what happens is it unzips itself so that you have two halves of DNA, and you can kind of imagine these two pieces floating apart. But since we know that all of the thymines always connect to an adenine, and all of the cytosines connect to a guanine, and the guanines to the cytosine, and so on, once the thing unzips and you have two separate halves, then new molecules can come in and connect to the connection points and make two completely new double helix twisted pairs. I'm going to say that one more time because even though I'm not going to explain the copying process in detail here, this is the core of why DNA is the way it is. Because the hydrogen bonds in the middle are just strong enough to hold it together, but they can be weakened during the copying process so the molecule can split apart. Once it splits apart, you have the base pairs on one half, and the connection point is now severed, and then you have the base pairs on the other half. Now you have two freely floating, independent half DNA molecules, but then new nucleotide bases can come in and connect to the attach points, because remember, A's always get connected to T's, T's to A's, A's to T's, and C's and G's always get connected together. So once you have two halves of DNA, new nucleotides can come in and build the other half of the DNA helix, and now you have two completely independent double helix DNA molecules with a complete copy of the original, because it unzips and then the new ones can only go into this pattern with how the nucleotides are connected in there. So the question is, and this was a question for many, many, many years, how do these nucleotides, why are they bonded? Why are the A's only allowed to connect to the T's? And why are the C's only allowed to connect to the G's? We're going to talk about that on the next slide. We are going to get there. But for right now, getting back to the Human Genome Project, the goal of the project was to read an entire DNA molecule from one end all the way to the other. Here I've only got maybe 20 or 25 of these base pairs here. It's called a base pair when they're paired up like this in the completed molecule. What the Human Genome Project wanted to do is read all of the letters from one end of the DNA molecule to the other end of the DNA molecule, right? 3 billion of these base pairs, A, T, T, A, T, C, G, A, T, T, C, C, G, A, T, C. You could see 3 billion of them throughout the whole molecule. It took over a decade for that research to actually occur. That's what the goal of the project was. Once we know what the code is, then we can understand diseases better to help us all. So now what we want to do is finally answer the question. In the DNA molecule, why does the adenine only connect to the thymine? Why does the guanine only connect to the cytosine? Let's go to the more detailed drawing. We'll come back to this, but we'll come to the more detailed drawing because I think it's easier to understand right here. Here is DNA in a little more detail. I told you education is a series of revealing layers, right? At first, we started with the simplest cartoon layer and said it's a double helix with some colored things in there. And then we said, okay, same thing here, but these colored things are really these complex carbon rings with all this stuff hanging onto it. Okay, that's a little deeper. Then we reveal another level deeper. The double helix phosphate backbone, notice yellow is phosphorus. You can see phosphorus all along the double helix, along the kind of like a ladder. If it was a ladder, it would be the sides of the ladder. We have the phosphorus there. And in the center is where the A's and the T's and the C's and the G's lie, as in the previous diagram. So we have kind of a inset here. You can see thymine connected to adenine, and this is the structure of thymine, and this is the structure of adenine, and this is the structure of cytosine, and this is the structure of guanine, right? So remember, A to T, over here, there was really 2 connection points between the 2 molecules, and C to G, there was like 3 connection points, 3 dotted lines here. So there's C to G, 3 connection points, and then 2 connection points here. So this drawing literally is the same as the previous drawing. It's just all of those carbons I told you about that were in here and the little kinks. These are all carbons right here. Now they're drawn explicitly as balls, but this is the same thing. Now what we want to do is zoom in on this. The A's, the T's, the C's, and the G's. Let's see how it works. This is a blow up of what we just saw right here. T and A and C connected to G, and you can see it a little more clearly. Now, couple of things. We need to figure out, why does adenine always connect to thymine? So let's look at this. The blue one here, or I guess the deep purple one here, if you look here, is nitrogen. So I'm going to go over here and I'm going to label this nitrogen. And the white here is hydrogen. We saw that on the other drawing there as well. So that's that. And then here is hydrogen, like this. So nitrogen is bonded to hydrogen. Okay, let's go down here. What's over here? What's red? Red is oxygen. So right here is oxygen. And white is hydrogen. So this right here is hydrogen. Okay? So let's just leave that on the board and try to understand, why does H want to connect or bond with N, and why does O want to connect or bond with H? To do that, I told you didn't need to understand it in chemistry, but I do need to reveal the periodic table. But I promise you, anyone can understand this. So if you're feeling anxious, don't worry. I promise I can make it clear. Please stick with me. Let's look at how a nitrogen and a hydrogen behave. On the periodic table, the hydrogen is in this first column, and the nitrogen is over here. Now, a lot of people think the periodic table is just a table, but actually it's organized that way for a reason. The periodic table, one main reason, is it tells us how many electrons an atom really wants to have when it reacts with something. And when I say it wants to have electrons, these elements, they don't have feelings, they don't have wants, okay? But the reason that they like to get electrons is because there's an electric force pulling them in. And the way you read the table is, anything over on the right-hand side, like when you point your finger at N, and you have 1 square, 2 squares, 3 squares over to the last column, this is how many electrons nitrogen wants to have in order to be most stable. So 1, 2, 3. Nitrogen wants to accept 3 electrons in order to be most stable. In chemistry class, we learn why nitrogen really wants the 3 electrons, but for now, just understand the table is organized to tell you that. Oxygen, it's 2 spots away. Oxygen wants to accept or take 1 more electron away, or to take 1 more electron to be stable. Fluorine wants only 1 more electron. You can pick anything on here. You know, carbon generally likes 1, 2, 3, 4 electrons to be stable. Sulfur likes 2 electrons to be stable. Selenium likes 2 electrons to be stable. Bromine likes 1 electron to be stable. That's how we read the table. So go back with me to nitrogen. Nitrogen likes how many? 1, 2, 3 electrons to be stable. Hydrogen, if you kind of think of it as wanting to accept another electron, wants to accept 1 more electron to look and be more stable like helium. In the periodic table, this last column here are the most stable elements in the universe. By stable, I mean they don't want to react. And they don't want to react, we learn in chemistry, because of the way the electrons are orbiting, and basically there's no force to pull any more electrons in. We'll get to the details of that in a chemistry class, but they're stable because there's no additional force from the nucleus to pull any more electrons in, essentially. So, back up to 100,000 feet. Hydrogen wants to accept 1 more electron when it bonds, and nitrogen wants to accept 3 more electrons when it bonds. So here we have a molecule where there's a hydrogen getting very, very close to a nitrogen. This nitrogen is connected to carbon here, getting 1 electron from carbon. Here it's getting another electron from this carbon, and so because it's only connected to 2 carbons right here, notice this gray one's carbon. If this is nitrogen, it's going to get 1 electron from carbon, 1 electron from carbon, that's 2. It needs a 3rd electron. Where is it going to get it from? It's going to get it from hydrogen. The hydrogen also has an electron around it, but remember, hydrogen itself wants to accept an electron. So when it bonds, hydrogen gets 1 additional electron from the nitrogen, and the nitrogen gets 1 additional electron from the hydrogen, because these are all atoms and they all have electrons around the nucleus there. So because nitrogen wants 3 and hydrogen wants 1 more, these are going to pair up, because in the adenine molecule right here, in the adenine molecule, it's already getting 2 electrons from the carbons. The 3rd one, it'll just share with the hydrogen, and the hydrogen will get the 1 more it wants from the nitrogen, so they're both happy. They're both happy because hydrogen wants 1 more and nitrogen wants 1 more because it already has 2 here, so they bond right there at that hydrogen bonding point. Okay, now let's come down here. Here's oxygen bonding with hydrogen. Oxygen wants 2 additional electrons, but hydrogen only wants 1 more. But if we go in here, this oxygen is connected to a carbon. It's getting and sharing an electron from this bond. It's getting 1 here, but it wants 2. It gets the 2nd one from the hydrogen. the hydrogen is getting one more from the oxygen, so they bond together. So you see, the reason thymine always wants to bond with adenine is because when you look at the connection points, those atoms are in the most stable configuration when they're bonding with hydrogen, because each side of the DNA is getting the exact amount of electrons that it wants. This is why thymine is never going to bond with guanine. First of all, look at the number of connection points. Right? Thymine has basically 2 connection points, but guanine has 3 connection points. So it's like a lock and a key. You have 2 connection points here and 3 connection points here. It's not going to bond because the connection points, they don't line up. So there's a physical aspect to it because you have to have the same number of connection points, but also the atoms don't even, they don't fit together in the way that they do right here. We have 2 connection points. Each connection point is happiest when it's bonded like this. Okay? Now let's go down and look at this molecule. We can understand it in the same way. This is cytosine and this is guanine. Okay, let's look at that one. I'll do that one in a different color. Now what we have here, this is oxygen right here. This is oxygen, and this is hydrogen right here. And right here, this is nitrogen. So I'm just going to put, I think, an N right here, and this is hydrogen right here. And then this is hydrogen right here, and this is oxygen right here. Now we already established that oxygen wants 2 electrons. It wants 2 electrons. So how's it going to get 2 electrons? It's going to share one of them with the carbon. It wants one more. It gets it from the hydrogen, but the hydrogen also wants one more, and it gets it from the oxygen. So they're sharing a pair of electrons at the connection point. Basically, every connection point here is like 2 electrons, and they're being shared on both sides. So the hydrogen is happy, and the oxygen is also happy. Nitrogen, we just said it wants 3 electrons. It's getting 1 from here, 1 from this carbon. It wants 1 more. It gets it from the hydrogen. The hydrogen also wants 1 more, and it's getting it from the carbon. So this is a stable bond. Same thing happening here. You have hydrogen here and oxygen. It's getting, oxygen wants how many? 2 electrons. It's getting 1 from the carbon and 1 from the hydrogen. The hydrogen wants 1 more, and it's getting it from the oxygen. So, you know, when I first learned about DNA, it's just told to you that A's and T's go together and C's and G's go together. It's told to you. But because usually books don't want to get too deep in the weeds too early on, they don't want to tell you exactly why. They don't want to draw this mess because it's just easier to tell you they bond together. This is why it bonds together, and it's critical to understanding DNA. Because when a DNA unzippers and becomes 2 halves, the only way to replicate the other half of the side is if, since you know the A's goes with T's and the T's goes with A's, if it unzippers over here, and you have a T hanging by itself, then another nucleotide will float in and connect to the T. An A would be what would come in, an adenine would come in and connect with the thymine right there, because that's the only nucleotide that can bond here. And if you were hanging off with a cytosine here, only a guanine could float in there, and then when the thing is reassembled, you're going to have an exact replica of the other half that split apart as well. That's how DNA replicates. Now, when you consider that every DNA molecule is about 3 billion with a B base pairs, 2 things should become apparent. Since we're dividing ourselves all the time, this copy process must be really, really good. And it is really good. But there are errors that creep up in your DNA replication. We call that cancer when we get older. Cancer is caused by lots of reasons, but it's copy errors in DNA. Now, our cells have lots of mechanisms to repair the damage if we accidentally copy a molecule incorrectly or a nucleotide incorrectly, and we're always repairing our DNA internally to our cells. That's happening all the time. But as we get older, the repairing process doesn't work as well, and so you eventually get errors in your DNA, and it causes diseases. So the Human Genome Project was trying to read these, all of these nucleotide base pairs from the beginning to the end of the DNA molecule, all of the A's and the T's and the C's and the G's, to understand how this thing is actually organized and how the genes are set up along the length of the DNA so that we can understand how diseases, how genetic diseases actually work. While the Human Genome Project identified and mapped the locations of all of the genes in the genome, it's important to note that not all of the functions of these genes are fully understood. The identification of genes in the genome was based on the sequence of the base pairs in the DNA, but determining the function of these genes requires further research. There are several reasons why we may know the genes in the human genome but not their function. One reason is that some genes may be involved in very complex functions or processes that are not yet fully understood. Another reason is that some genes may have multiple functions, and it may be difficult to determine which function is the most important or relevant in a given context. Finally, some genes may be involved in processes that are not yet fully understood or recognized, and it may take time for these to be discovered. So what I'm trying to say here is that the human genome did map the entire genome top to bottom. All the A's, all the T's, all the G's, all the C's, okay, that map in the genome. But it doesn't mean we understand everything about DNA and how it works, because we understand the code, but we don't exactly know what all the genes do because we don't know exactly how all the genes are interrelated. So a gene is a sequence or a subset of these letters. If you could read all 3 billion base pairs, then if you divided them into different length segments, we would call those genes. So different patterns of letters or different nucleotide bases is what we would call a gene. But the problem is we don't always know what these genes actually do. We don't know which genes control eye color, for instance, exactly, and which inherited genes control, the thickness of your hair. Because a lot of times the genes, it's not just one single gene that actually affects the characteristics of your body. Many times it's multiple genes. In fact, modern research is showing that a lot of the things in our body, like physical characteristics and things like that, may be controlled by dozens or even hundreds of different genes. So it's not just knowing the letters, it's knowing what the letters do, what the nucleotide bases do, and how everything interacts to produce some external characteristic of our body. There's one thing I haven't gotten into, we'll get into, we study biochemistry one day, is that these sequence of letters in the DNA molecule, the nucleotide bases, what they're coded for, what these codes represent in the genes is usually to construct a protein. A protein is a molecule in the human body that gets most of the work done in the cells. So these genes on the DNA are basically constructed to produce proteins in the body. So if you're looking at like skin color or hair thickness or something like that, or heart failure or things like this, or heart characteristics, they're usually controlled by multiple different genes that make different proteins. And some of the genes are only triggered in some people and not others based on other chemicals as well. So it's not just as simple as, oh, I know the sequence, now I know everything about it. It's a good first step. We have to know that. But in order to actually know what turns on and off different diseases and things, we have to do further research to identify how many genes are involved and how they're interrelated to affect whatever the disease or the characteristic that we're trying to study. While we now have a fairly comprehensive understanding of the genes in the human genome and their locations, there's still much that we do not yet know about the function of these genes and the ways in which they contribute to the characteristics and traits of individuals. Further research is needed to fully understand the role of the genes in the human genome and how they contribute to the complex traits and characteristics of humans. Just to expand on this a bit, we'll go back to our little simple cartoon, and we've dived in there, and we kind of know in general how in the details here, how the nucleotide bases are connected. But let's zoom out here, and let's say that part of the human genome reads, some sequence, right? So adenine, cytosine, guanine, thymine, and cytosine, let's say, right? A, T, C, and G. So there's only 4 different bases, but they're connected in different order, right? And we call this thing a gene. Now I'm leaving out a lot of details because the structure of DNA actually has markers in the DNA that denote the genes. So it's not just a random sequence of letters. There's markers in the DNA that tell us where the genes are. So we may have mapped that this is a gene. 2 adenines, and this is like 1/2 of the DNA here. 2 adenines, cytosine, guanine, thymine, cytosine. Again, 1/2 of the DNA. These are connected to the other side. The A would connect to a T here, thymine. The C would connect to a G, guanine. The guanine would connect to a cytosine. The thymine would connect to adenine, and this cytosine would connect to a guanine. So this is like 1/2 of the DNA. A, C, G, T, C, and be connected to its kind of its partner side on the other side here. So we denote this as a gene. Great. What does that mean? I don't know. Does it control how long your toenails grow? Does it control how good your cells repair themselves? Because the repair process is governed by DNA as well, right? Does it, or is there a problem with your DNA causing a genetic disease? Maybe this C in this gene really needs to be a T in order to function properly and make the correct protein. So maybe the protein that governs something in your body is not being made in the right amount, and then you're always sick from birth from that. Or just an example. There are lots of genetic diseases. Okay, so part of the Human Genome Project is obviously mapping everything, understanding where the genes are. But now we have to take that information and figure out how the genes interplay with each other. Because maybe there's a gene here, but then there's a gene 6,000 base pairs that way, which is another gene, and this gene makes a specific protein, and the other gene down there makes another protein, and maybe it takes both proteins together to have pigment in your skin properly, let's say. Or maybe 6 genes. Or it may be 150 genes that make different proteins in different amounts to make your hair grow properly or something. So the point is, the characteristics of our body and diseases are often governed by multiple genes. So it's not as simple as knowing the sequence. We have to do research to know how the different genes are actually controlling everything in the body. The project employed a number of different techniques and technologies in order to achieve its goals. One of the key techniques used was DNA sequencing, which involves determining the order of the base pairs in a piece of DNA. This was accomplished using automated DNA sequencers, which are machines that can read the sequence of base pairs in a DNA molecule. Another important technique used was genome mapping, which involves creating a map of the locations of the genes on a chromosome. This was done using a number of different methods, including restriction fragment length polymorphism analysis and fluorescent in situ hybridization. These sequence technologies were made more affordable and rapidly increased in speed due to the Human Genome Project. In addition to these techniques, the project also made use of a number of computational tools and resources in order to analyze and interpret the data generated by the project. These included databases, software programs, and algorithms that were used to analyze and compare the DNA sequences obtained for the project. So what I'd like to add to that is just to say that not only did they have to sequence the DNA, but they had to invent a lot of computational tools, supercomputers, other computer systems and architectures to store the information, share that information with the other research institutions so they weren't duplicating effort, because there's 3 billion with a B base pairs, right? So they wanted to divide the work up, they wanted to make sure it was all correct, and we had to use computational tools that were also becoming very powerful in the time of this project in order to make the project a success. The Human Genome Project has had a major impact on the field of genetics and has led to numerous medical and scientific breakthroughs. The project has enabled researchers to identify the genetic basis of many inherited diseases such as sickle cell anemia, cystic fibrosis, and Huntington's disease. It's also led to the development of new treatments for these diseases as well as for many other inherited conditions. Now I want to talk about this for a second because this is one of the main reasons why we did this project. Many diseases are inherited or passed down through our genetic makeup. If we can understand the genetic makeup of humans, then we can understand ahead of time when someone's going to get sick and maybe even have a way to repair the DNA. And that's kind of the cutting edge of research today. So just one example, but keep in mind there are many others, is what we call sickle cell anemia. So in your blood, there are red blood cells. Now, I'm not a good artist, right? I'm not a good artist at all, but a red blood cell is essentially a circular cell. It's got like a little depression in the center, and the red blood cells are in your blood and they're bonded to the oxygen, carrying the oxygen all throughout your body. Now, in a person with sickle cell anemia, because of some of the genes in their DNA are not correct, then the proteins that are created to make the red blood cells are not correct. And so instead of making beautiful circular round donut-shaped red blood cells, they're these kind of like these moon-shaped blood cells. So they're not circular at all, they're kind of like these weird little half circles or quarter circles. Now, these red blood cells, when they're kind of malformed like this, cause a number of problems. The first one is that the red blood cell can't transport the right amount of oxygen in your blood, so you can never get enough oxygen. That's obviously bad, right? But also, because these things are malformed, they actually can clog up your bloodstream, and they can cause clotting problems and clogging problems in your circulatory system. And so sickle cell anemia is a genetic disease that's obviously affects a lot of people, and we have treatments for it, but as far as a cure, the way we're looking at it today is by actually changing the DNA to be correct. That's called gene editing or genetic therapy or genetic medicines. So step one is to figure out, do you have sickle cell anemia? What is your DNA doing? Why are you afflicted with this? And the best way to treat it is not by just treating the symptoms. It's to try to go in there and fix the DNA. If we know that one or more of these nucleotide bases in a certain gene is wrong, and if we can create a medicine that can go in and change the DNA to the correct sequence, then we can cure the genetic disease. Now, there are many, many genetic diseases. That was just one, sickle cell anemia, but there are many, many others. Many of the other genetic diseases are not just affected by one gene, affected by many genes. So we have to understand how the genes are working together to cause the problem so that we can determine what to fix. And it's almost like we're doing it blind because we know the sequence, but we have no idea how the interplay of the genes unless we do further research. So that is what a lot of biotechnology research is doing now, is figuring out how these genes work together to make the body function so that whenever the body's not functioning right, we can figure out how to repair it. One way in which the project has reduced cost for DNA sequencing is by developing more efficient and cost-effective technologies for DNA sequencers. Prior to the completion of the Human Genome Project, DNA sequencing was a labor-intensive and expensive process. The project played a key role in developing the automated DNA sequencers, which are machines that can read the sequence of base pairs in a DNA molecule much more quickly and accurately than was previously possible. These sequencers have greatly reduced the cost and time required for DNA sequencing, making it more accessible and affordable. Now, just to add to that, when the project started in 2001, the cost, or the estimated cost, to sequence an entire genome, an individual genome from a person, was $1 million per genome. $1 million with an M dollars to sequence a genome, right? But as the project went on year after year, the sequencers get better and better and better because we're throwing all of this funding and this money into accelerating, the research. And so by the end of the project, the human genome was averaging somewhere around $900 or $850 or $900 for a genome to be sequenced. So let me say that one more time. At the beginning of the project, it was costing about $1 million to sequence, if you project it out, because what was happening in the beginning, it was taking so long to sequence individual base pairs. If you multiply that by 3 billion base pairs, they estimated it would be about $1 million per genome once it was mass produced and kind of mass applied. But then toward the end of the project, nowadays, it's around $900 per genome. And the goal with the companies that are making these sequencers is to get it down below $100. So we're going to talk more about this in a few minutes, but the goal here is to make it so cheap and so easy to sequence your DNA that everyone just gets it done, because it'll be a great way for us to just make sure that we don't have any serious genetic diseases. And the only way that that kind of cost reduction can happen is by dumping a lot of government funding into a project, driving the cost down year after year. In fact, I remember reading articles and seeing interviews at the beginning of the Human Genome Project where some scientists were actually saying that the project would never get completed because the cost would be so high to do this at the rate at which it was going that they were saying it would take 50 years and so much money that it would never get completed. But one thing they couldn't plan for was the cost reduction of the sequencing equipment, and that only happens when you have lots of research funding being pumped into researching new ways in order to develop these technologies. In addition to reducing costs for DNA sequencing, the Human Genome Project has also contributed to the development of personalized medicine, which is a medical approach that involves the use of genetic information to tailor treatment plans to the specific needs of individual patients. The project has enabled researchers to identify the genetic basis of many inherited diseases, which has led to the development of new diagnostic tests and therapies that are based on a person's individual genetic makeup. This has the potential to improve the effectiveness of medical treatments and to reduce cost by targeting treatments more precisely to the needs of individual patients. So this was sort of the unwritten goal of the whole thing. Obviously, they wanted to map the entire human genome, but we wanted to make it cheap enough so that everybody could individually get genetically mapped if they so chose. Now, we're not forcing people to do this. It's not something that we're trying to make it happen, but make everybody compulsorily do this. But if you can imagine a future where the cost of sequencing your DNA is so cheap that it's almost like just getting some cough drops from the drugstore or just some simple over-the-counter medicine, maybe at birth, if you elect to do so, you're just sequenced automatically at birth, just like we do a lot of screening for other diseases at birth. And then we can look in the DNA and see what genetic diseases do you have. And then when gene therapies come online, once we understand how to repair and correct these kinds of errors in the DNA, then, of course, with your permission, then we can obviously effect a repair for a life-threatening illness. Right? Of course, there's other questions, you know. Some people are like, well, maybe I can make designer babies, or maybe I can make the perfect human. That's not what I'm advocating for here. I'm simply saying there are a lot of diseases that are in our genetic code. It would be nice to know about that ahead of time, and if we have the technology to effect a repair, I certainly would want that option. Overall, the Human Genome Project has been a major scientific achievement that has had a profound impact on the field of genetics and on society as a whole. It has led to numerous medical and scientific breakthroughs and has opened up possibilities for the understanding and treatment of inherited diseases. While the project has also raised important social and ethical issues, it has also had significant economic impact and has helped to drive the growth of new industries and technologies. The project has led to the development of new diagnostic tests and therapies that are based on a person's individual genetic makeup, and has enabled doctors to more accurately predict a person's risk of developing certain diseases and to tailor treatment plans accordingly. So in summary, I hope you've enjoyed this overview of the Human Genome Project. I hope you've especially appreciated the time that we spent talking about the DNA molecule. I want you to come away from this understanding something more than you knew when you started. But I also want you to know that what I've talked about here is not the end of the story. There are lots of details of DNA that we just do not understand. We don't know certain things about the replication process. We don't understand the intricacies of how the genes combine and work together to actually affect things like your fingers and your skin and other genetic diseases. So this was a very first step. It's like it's basically like you go to an alien planet and there's a computer terminal sitting in the alien spaceship, and you get a copy of the computer code, right? So you get a copy of the computer code, you know in your hand exactly how that spaceship runs because all the computer code is in your hand. However, it's completely foreign code. You don't know what the code exactly means, but everything necessary to run that ship is in your hand. Now what you have to do is figure out through trial and error how that code functions. How does it make us grow? How does it make us age? How does it make us die? How come some people seem to live longer and some people have genetic diseases and don't live as long? Why does cancer affect certain populations more than others? How does cancer work? How can we prevent cancer, right? Just one or two or three of many, many thousands of questions you could raise. But the first step was mapping the human genome, understanding how DNA works. And that's what the project was. It truly was one of the most important scientific projects of the last 100 years.